Glycosylation is the reaction in which a carbohydrate, that is a glycosyl donor, is attached to a hydroxyl or other functional group of another molecule. In biology, glycosylation mainly refers in particular to the enzymatic process that attaches glycans to proteins, lipids, or other organic molecules. This enzymatic process produces one of the fundamental biopolymers found in cells. Glycosylation is a form of co-translational and post-translational modification. Glycans serve a variety of structural and functional roles in membrane and secreted proteins. The majority of proteins synthesized in the rough year undergo glycosylation. It is an enzyme-directed site-specific process, as opposed to the non-enzymatic chemical reaction of glycation. Glycosylation is also present in the cytoplasm and nucleus as the O-GlcNAc modification. Aglycosylation is a feature of engineered antibodies to bypass glycosylation. 5 classes of glycans are produced, unlinked glycans attached to a nitrogen of asparagine or arginine side chains. Unlinked glycosylation requires participation of a special lipid called aryl phosphate. O-linked glycans attach to the hydroxyl oxygen of serine, threonine, tyrosine, hydroxylysine, or hydroxyproline side chains, or to oxygens on lipids such as ceramide, phosphoglycans linked through the phosphate of a phosphoserine. C-linked glycans, a rare form of glycosylation where a sugar is added to a carbon on a tryptophan side chain, glycation, which is the addition of a GPI anchor that links proteins to lipids through glycan linkages. Purpose: glycosylation is the process by which a sugar is covalently attached to a target protein. This modification serves various functions. For instance, some proteins do not fold correctly unless they are glycosylated. In other cases, proteins are not stable unless they contain polysaccharides linked to the amide nitrogen of certain asparagens. Experiments have shown that glycosylation in this case is not a strict requirement for proper folding, but the unglycosylated protein degrades quickly. Glycosylation also plays a role in cell-cell adhesion via sugar-binding proteins called lectins, which recognize specific carbohydrate moieties. Glycoprotein diversity: glycosylation increases diversity in the proteome, because almost every aspect of glycosylation can be modified, including glycosidic bond the site of glycan linkage, glycan composition the types of sugars that are linked to a given protein, glycan structure can be unbranched or branched chains of sugars, glycan length can be short or long chain oligosaccharides. Mechanisms: there are various mechanisms for glycosylation, although most share several common features. Glycosylation, unlike glycation, is an enzymatic process. Indeed, glycosylation is thought to be the most complex post-translational modification, because of the large number of enzymatic steps involved. The donor molecule is often an activated nucleotide sugar. The process is non-templated. Instead, the cell relies on segregating enzymes into different cellular compartments. Therefore, glycosylation is a site-specific modification. Types of glycosylation: unlinked glycosylation. Unlinked glycosylation is the most common type of glycosidic bond and is important for the folding of some eukaryotic proteins and for cell-cell and cell extracellular matrix attachment. The N-linked glycosylation process occurs in eukaryotes in the lumen of the endoplasmic reticulum and widely in archaea, but very rarely in bacteria. O-linked glycosylation. O-linked glycosylation is a form of glycosylation that occurs in eukaryotes in the Golgi apparatus, but also occurs in archaea in bacteria. Phosphoserine glycosylation, xylose, fucose, mannose, and glycNAc phosphoserine glycans have been reported in the literature. Fucose and glycNAc have been found only in Dictyostelium discoideum, mannose in Leishmania mexicana, and xylose in Trypanosoma cruzi. Mannose has recently been reported in a vertebrate, the mouse, Mus musculus, on the cell surface laminin receptor alpha-dystroglycan 4. It has been suggested this rare finding may be linked to the fact that alpha-dystroglycan is highly conserved from lower vertebrates to mammals. C. mannosylation, A mannose sugar is added to the first tryptophan residue in the sequence WXXW. Thrombospondins are one of the most commonly C-modified proteins, although this form of glycosylation appears elsewhere as well. C-mannosylation is unusual because the sugar is linked to a carbon rather than a reactive atom such as nitrogen or oxygen. Recently, the first crystal structure of a protein containing this type of glycosylation has been determined, that of human complement component 8, PDBID30JY. Formation of GPI anchors. A special form of glycosylation is the formation of a GPI anchor. In this kind of glycosylation, a protein is attached to a lipid anchor, via a glycan chain. Clinical, over 40 disorders of glycosylation have been reported in humans. These can be divided into 4 groups: disorders of protein N-glycosylation, disorders of protein O-glycosylation, disorders of lipid glycosylation and disorders of other glycosylation pathways and of multiple glycosylation pathways. No effective treatment is known for any of these disorders. 80% of these affect the nervous system. See also, glycorandomization, glycation, advanced glycation end product, chemical glycosylation, fucosylation. References. External links: GlycoEPA in silico platform for prediction of N, O and C glycosides and eukaryotic protein sequences PLOS18, 6, E67008, online textbook of glycobiology with chapters about glycosylation, glyprot, in silico N-glycosylation of proteins on the web, NetNGLYC. The NetNGLYC server predicts N-glycosylation sites in human proteins using artificial neural networks that examine the sequence context of AS and XOS-THR sequence. Supplementary material of the book The Sugar Code, additional information on glycosylation and figures.